Termination phase of translation in eukaryotes. This phase again has several mechanistic similarities with the prokaryotic system. So let us look at the learning outcomes of this session. Termination of translation occurs when one of the stop codons is present on the A site, and this A site, which has the stop codon, against the stop codon there is no cognate tRNA present. Therefore, no tRNA will come and associate, or basically the elongation factor 1A will not bring any tRNA to the A site, as there are no tRNA that would bind to the stop codons. The release factors ERF1 and ERF3 form a quaternary ERF1 ERF3 Mg2 plus GTP complex, coordinating the termination initiation. The GTP binding, this GTP that is bound to this complex, and its utilization regulates the termination. The polypeptide is released from the ribosome due to hydrolysis at the acyl bond between the tRNA and the polypeptide. The disassembly of the ATS and other factors is the onset of recycling of translation, and this is the concept of what is called as the ribosome cycle. So the termination phase of the translation in eukaryotes is looking at two things. One is termination by removing or releasing the polypeptide, and second, by disassembly of the entire ATS complex. Now, let us understand the process of termination of translation by looking at it in more detail. So consider the fact that after elongation, you have a tRNA associated with the polypeptide at the P site. The E site would have had a deacylated tRNA which slowly moves away from the complex, and the A site is free for another tRNA to be brought in. But suppose the A site now has at the decoding center the stop codons either UAA, UGA or UAG, then these codons can be decoded by a ternary complex. And the ternary complex comprises the ERF1, this is the release factor 1, ERF3, release factor 3, which is associated with a GTP molecule. So these three constituents form what is called as the ternary complex. Now the ERF1 belongs to class 1 release factor, and the ERF3 belongs to the class 2 release factor. This ERF3 has an inherent GTPase activity, and therefore the presence of GTP and the GTPase is a point or is a contention that the ERF3 plays a role in regulation. The ERF1 recognizes the stop codon with high efficiency, and it initiates the hydrolysis of the polypeptide, which means that ERF1 is responsible for catalytically cleaving the polypeptide from the associated tRNA. And this catalysis takes place at the peptidyl transferase center itself. The class 2 release factor does not recognize the stop codons. However, it has the most important role of stimulating the class 1 factor, that is, it stimulates the ERF1 to carry out the hydrolysis and release the polypeptide, and not just that, but also recognize the stop codons. What is very important to note is that it is the ERF3 GTP complex that is actually delivering the ERF1 to the A site, such that it binds to the stop codons. And because it has the inherent GTPase activity, it confers GTP dependency on the entire process. Now, when you look at the structure of ERF1, that is release factor 1, what is very clear is that the ERF1 biomimics or is a biomimicry of the tRNA itself. So you can see that the domain 1 has anticodon like site, so it can bind to the codons, which stop codons. So it has a set of 3 amino acid that acts as an anticodon. So you can see how it is similar to the anticodon arm of the tRNA. It has a GGQ, which is highly conserved at the N-terminal region, and this is very similar to the CCA conserved sequence at the acceptor arm of the tRNA. And of course, you have this domain 3, which is kind of similar to the T-Cy stem of the tRNA. So effectively what we understand therefore is that the ERF1 resembles tRNA, showing the biomimicry phenomenon. Now of course, as mentioned, various studies have shown that the GGQ, which is a highly conserved motif in the ERF1, plays an important role or is responsible for hydrolysis of the acyl bond that is present between the tRNA and the polypeptide chain at the P site. The ERF3 has been observed we know that ERF3 delivers ERF1 to the A site, but the effective binding of the ERF3 to the ERF1 ensures that the GGQ is positioned at the peptidyl transferase center of the large subunit for catalysis. So ERF3 therefore is able to stimulate the ERF1 to release the polypeptide from the ribosome complex. Now, when the entire ribosome complex was reconstituted in vitro and through cryo-electron microscopy, the structure was studied, it was found that there were two distinct complexes present during the termination phase. One is what is pre-termination. So when you call it pre-termination is before the release of the polypeptide chain, and the post-termination or termination itself is the release of the polypeptide chain. So the pre-termination complex comprises ERF1, ERF3 and GTP. And you can see over here how at the P site you have the tRNA with the which is acylated to the polypeptide chain. At the E site you have the deacylated tRNA which moves away from the E site, and at the A site, which has the stop codon, the ERF3 is positioning the ERF1 onto the A site, which means the ERF1 is able to bind to the stop codon that is present at the A site, and this is thanks to the ERF3 being able to position it such a way that ERF1 recognizes and interacts with the stop codon. The moment you have the ERF1 interacting with the stop codon, what is observed is that there is a slight conformational change because there is an interaction over here, and that conformational change leads to ERF3 GTPase activity getting active, and therefore ERF3 becomes ERF3 GDP. As ERF3 GDP, it has less affinity to bind to the ERF1 and it is removed. But in place of the ERF3, you have another molecule that comes in, which is ABCE, that is ATP binding cassette element 1. And this comes and positions or binds to the ERF1. So the moment you have the ABCE1 binding to the ERF1, you can very clearly see how the conformation of the ERF1 is changing. So the ERF1 N-terminal domain has the NIKS motif, which is acting as the anticodon, and you have the ERF1 central domain containing the GGQ, and this GGQ is positioned at the peptidyl transferase center. So basically the reaction that happens at the peptidyl transferase center during elongation is a transesterification reaction where the tRNA with the amino acid present at the A site is able to carry out a nucleophilic attack on the polypeptide that is acylated to the tRNA at the P site with the involvement of the 28S rRNA in the peptidyl transferase center, this bond is to be broken and you have a bond formed between CO and NH. So therefore, this amino acid now is in a peptide bond with this polypeptide. And this amino acid the polypeptide has been transferred to the tRNA at the A site. This is what happens during elongation phase. But what happens at the termination phase is that the A site obviously does not have a tRNA, but instead it has the ERF1. And the ERF3 ensures that the GGQ is positioned at the peptidyl transferase center. Now the glutamine has a CONH bond, a CONH functional group at the end of its side group, or the R group has the CONH, and this CONH2 is able to interact with the water molecule, and this is basically the electrons from the base in the 28S rRNA in the peptidyl transferase shifts its electron here, which activates the electronegative O of the water that carries out an attack on the acyl group of the tRNA at the P site, and because of that you have this acyl bond being broken and the polypeptide chain being released. So therefore, you have the peptidyl tRNA hydrolysis taking place because of the glutamine. Several studies have shown that if the glutamine is replaced by any other amino acid, one does not observe the peptidyl tRNA hydrolysis, and therefore termination of translation does not happen properly. That is why it is very important that these GGQ is highly conserved in the ERF1, and that is responsible for the termination process to occur. But what should be very clearly noted over here is that the ATP binding cassette element 1, when it binds to the ERF1, the conformation change of ERN1 is stabilized by the ABCE1 and the GGQ is positioned at the peptidyl transferase center. Now we look at the second aspect of termination of translation, and that is what is called as translation recycling, or what we call as ribosome recycling. So to look at what happens or what we understand by recycling is that when you have the initiation occurring, you have the ATS complex being formed, and due to the elongation factors, you have the polypeptide getting synthesized, and when it reaches termination, because of the RLI, this is the counterpart of ABCE1 in the yeast, because of that, you have the polypeptide getting released and the ABCE1 with its ATP bound to the ATS. Now, when it binds to the ATS, the conformation of the ATS changes, and that in turn activates the ABCE1 such that the NTP gets hydrolyzed to NDP. Now, the moment NTP gets hydrolyzed to NDP, energy is released, and that energy is basically enabling the dissociation of the entire complex. So, you can see how the 60S gets dislocated from the 40S, and you have the mRNA and gradually all the other constituents you have the mRNA still associated with the 40S. So this 40S can again go and bind to the start codon and initiate translation process again. So that is what is understood by ribosome recycling or translation recycling. Now to look at how the translation recycling is made feasible by the initiation factors itself, we look at this. So we know that the pre-initiation complex that is formed at the 5' cap of the mRNA is also able to bind to the poly A binding protein that in turn is associated with the 3' poly A tail of the mRNA. So you can see how this complex is not just associated with the 5' cap of the mRNA, but it is also associated with the end of the mRNA. So you can see how the mRNA is circularized, and the termination or the stop codon is very close to the start codon. Where this do not happen, you would have the start codon here and you've had the stop codon at the other end. But now because of the circularizing of the mRNA due to the binding of the initiation complex to both the 5' camp and the poly A tail, you have the stop codon very close to the start codon. So if something disassembles at this point, assembly at the start codon can be more easy because it is placed very close to each other. It is juxtapositioned very close to each other. So the APCE1, that is the ATP binding cassette element 1, is proposed to play a dual role. It helps in the disassembly of the ATS. So we already know that after the ERF1 is releasing the polypeptide and the ABCE1 gets activated such that its ATP is hydrolyzed to ADP, you can see how the disassembly of the entire ATS is taking place, but the ABCE1 is still associated with the 40S. So this ABCE1 recruits all the initiation factors and enables the formation of the pre-initiation complex again at such a way that the start codon is positioned at the P site. And then because of that, you would have initiation occurring, you would have the ATS complex forming, and then the starting of the translation takes place. So the ABCE1 is actually able to interact with 18S rRNA, which is present in the small subunit, and it can also interact with small subunit and large subunit proteins, and on hydrolysis of ATP is able to dissociate the subunits. So ABCE1 is helping translation recycling by not just disassembling ATS at the termination phase, but also formation of the pre-initiation complex by recruiting the initiation factors in the initiation phase of elongation. So let us make the conclusions. As elongation progresses, when the ribosome reaches a point where the stop codon is positioned at A site, the class 1 release factor ERF1 is able to recognize and interact with the stop codons. The ERF1 forms a ternary complex with ERF3, which is a class 2 release factor, and this in turn is a GTPase. The ERF3 aids the ERF1 in catalysis, with GGQ and in ERF1 responsible for catalysis. So this GGQ is a highly conserved sequence in the ERF1. The ERF3 hydrolyzes the GTP and gets released, with APCE1 binding to ERF1. This changes the conformation of ERF1 such that it is ready for hydrolysis and release of the polypeptide. The ABCE1 dissociates the assembled ATS and reinitiates translation by recruiting the initiation factors. So this is what we understand by ribosome recycling. Thus, the termination phase of translation in eukaryotes is subdivided into two parts, with one part seeing the release of the polypeptide and the second part the disassembly to enable ribosome recycling. Thank you.